Having me. I think this is a wonderful opportunity. We do quite a bit of research on campus focused on aging, and I think it's just a wonderful opportunity to make sure we are communicating with that with our greater Madison community. And so we'll talk a little bit about this geroscience hypothesis that is out in our field, and how we are trying to test that hypothesis with a couple of studies that we have ongoing. I'll talk about those studies and then would love to address any sort of questions or comments at the end. And so one of the reasons we're so interested in aging research is that with increasing age, there is an increased risk of one or more chronic conditions. And so I know this paints a little bit of maybe of a dire picture, but one of the interests for us is how can we slow the development or delay the development of those age-related chronic conditions so that we can continue to live a vigorous and healthy lifestyle. And so with this kind of schematic that's shown here, it's not just one usually chronic condition. As 50% of adults over this age of 65 generally have 2 or more chronic conditions. And so that suggests to us that aging is a risk factor for a lot of these different conditions. And if we can kind of intervene on some of the fundamental properties that we think are important for aging, then perhaps we might have an opportunity to age more gracefully. Additionally, we are about to embark in the first time in the United States history where adults over the age of 65 are going to outnumber children under the age of 18. So we have more and more older adults over the age of 65, and this is not only important from a personal standpoint, as we want our friends, our family, etc., to make sure that they're living a high quality of life, but also from the economic aspect from the country at large, as with this growing aging population and increased risk for diseases, this is also going to lead to Medicare expenditures well over $1 trillion. Luckily, there is this proof of concept that came out over 30 years ago now that actually indicates that aging and even the health span or the quality of life that different model species exhibit could be modifiable. And so this is a study from Cynthia Kenyon's lab, like I said, 30 years ago, and this is in worms, so take it for what it's worth. These are not humans. But what this shows is they were able to manipulate a gene called DAF2. And without getting into some of the nuance of that, it's a protein that's involved in the insulin signaling pathway. So they were able to manipulate this gene in worms, and they showed that the worms can actually live twice as long. Now, I'm not suggesting that this is necessarily feasible for humans, but it definitely suggests that that this is not as aging is not as static as we once maybe interpreted it to be. So perhaps it could be modifiable. Also, in subsequent studies, they were able to find that even functional deficits were delayed with manipulating this gene. So it's not only the amount of life lived, but there's also potential for the quality of life lived that this could be perhaps extended. And so that gets us to some of the objectives today. We've talked about how aging may be associated with different risks of chronic conditions, but we'll talk about this geroscience hypothesis. So we'll talk about that, and then what we use is a translational research approach. So we'll talk about what that actually means, and then we'll introduce some ideas in the field of why different compounds or drugs that could potentially modulate the biology of aging to extend healthy longevity. And so, without further ado, this is one of the kind of concepts that's been developed at the National Institutes of Aging, is this geroscience hypothesis. And so what this proposes is that we have a variety of different biological and metabolic functions of our cells and tissues throughout our body, and they're all different, they're all highly related. And so on the right here, with these aging biology, it shows different processes. They're highly connected, interrelated, and generally speaking, these start to change as we get older. And so they become a little bit more dysfunctional, and what that leads to is a loss of physiological function. So think about walking speed, think about your exercise capacity, etc. And so with this loss of physiology, this makes us more susceptible to the development of different age-related conditions. And so there's this connection of aging biology that becomes dysfunctional that makes us perhaps more susceptible to developing all of these different conditions that seem to be increased in prevalence with age. And so how we go about studying this is we use this translational geroscience approach. So it's really the combination of biology with gerontology. And so in our lab, we use tissues that are grown in culture dishes. We use different rodent models. We have a primate center here on campus that we are able to collaborate with. And then we also run human clinical trials. And the whole goal is that we can do a lot of different things in culture and in animal models that we can't necessarily do in humans, so that we can really truly appreciate the biology, which is very complex, and really understand different mechanisms of action, so that way we can help inform different human clinical trials to perhaps see if some of these different interventions can extend healthy longevity. And we also, our lab doesn't necessarily do this, but we also have great resources on campus that get out into the community. So if we are able to find anything that is positive or beneficial, hopefully we are able to disseminate that out to the community, much like today. And so there's this concept called health span. And so basically what this suggests is that across our lifespan, there's a period of time where we are free from chronic conditions, we are not slowed down, we don't have very many ailments, we are in a good quality of life. And then that's the subsequent second part of that is where eventually we may develop a condition, a disability, etc. And so our whole idea is that we want to try to extend and preserve that healthy or health span period of life and compress the time that was spent with trying to manage different diseases or mobility impairments. And even with that health span period, as we age, I think we can all appreciate that there's subtle declines in our function. Whether it's our slight increase in glucose that we may see in our lab reports, changes in our ability to do different activities of daily living, these generally will decline, but we're not necessarily impaired. We just start noticing these things. And these are risk factors for the development of subsequent disease. And so our idea is that we really want to intervene on and try to test the idea if we can use these different interventions, whether it's exercise, whether it's diet, or perhaps it's a drug compound. Can we intervene before the onset of these chronic conditions so that we can try to prevent them? And so we're going to focus on this drug called rapamycin. Most people may not have heard of it. It is in the social media spheres these days because it's a very kind of interesting topic. But rapamycin is an FDA-approved drug. It was approved first back in 1999. It's generally given to patients who are undergoing kidney transplant. It helps prevent the rejection of their transplant. And patients take anywhere from 2 to 10 milligrams of this drug per day, and that will be important as we move forward. Now, importantly, while it is FDA approved for selected indications, it's also involved in some cancer cocktail treatments, it's not approved for aging or age-related diseases. So this is not necessarily an endorsement to go out and try to start taking this compound on your own. But the way this drug works is it actually attenuates this protein called mTOR, which is actually named after the compound. So this drug was developed or found in the soil of Easter Island. It was an antifungal and antibacterial compound. And it actually was shown to inhibit this protein, and they named it after the drug itself. So it's the mechanistic target of rapamycin. And this signaling protein generally increases in most of our tissues across our body, and it's involved in regulating growth, proliferation, and metabolism. And so generally linked with dysregulation of these processes with aging. And so the question becomes, if we can attenuate this protein in the body, can we slow down the aging or the risk factors associated with aging? And you might be thinking, well, that's very ambitious of you to think that this could be possible. And I would tend to agree with you, but there is growing evidence to suggest that in multiple model systems, flies, worms, mice, and even some non-published data in monkeys or non-human primates, that this drug can extend lifespan, and it can delay several age-related pathologies that are commonly observed. And so the figure here is just one example. This is a project funded by the National Institute on Aging. It's a multi-site trial in mice to make sure that there's no site-specific, just kind of unique findings, that this is broadly generalizable. And what they show here is that rapamycin, in this case it's enriched in the diet, and there's increasing doses of rapamycin. So it's 4, 14, and 42. There are dose-dependent effects on lifespan extension in both male and female mice. And these aren't just your typical laboratory mice. These are what we call heterogeneous mice. So they're a mixed breed. So the idea is that hopefully this model this mouse model can better reflect the genetic diversity that we commonly see in people. And so this is just one example where there's dose-dependent effects. The greater amounts of rapamycin you take, or these mice would consume, the greater their lifespan extension. And so in addition to lifespan, there's been some studies, very few in humans, but and a lot in animals, and to show that there's improvements in physiological function. And so this first paper was in men, where it showed that rapamycin can improve glucose metabolism and insulin sensitivity. So these are generally thought to be positive benefits that would stave off type 2 diabetes. In mice, when rapamycin was given to mice in older age, started at older age, it actually reversed pre-existing cardiovascular dysfunction. So any sort of age-related deficit that was occurred, it was able to be rescued back towards its younger counterparts. In addition, there's been some data to suggest that rapamycin can also slow the loss of muscle mass and function, and therefore preserving mobility and activities of daily living, again, in mice. Rapamycin's also been shown to cross the blood-brain barrier, so it actually has been shown to improve blood flow to the brain. And blood flow is a known kind of risk factor for dementia and mild cognitive impairment. So the idea that inhibiting or attenuating mTOR with rapamycin in the brain, and it could restore blood flow, is very potentially advantageous to delaying the onset of any sort of cognitive impairments. And last, there's one study in humans. This is a 6-week trial where it found that a rapamycin analog was able to improve or rejuvenate aspects of the immune system. So it actually improved vaccine efficacy to the influenza vaccine. So these are all the good. This is all the good that we have. There's a lot of potential. There's a lot of enthusiasm because of these repeated results that have been shown over and over in multiple different laboratories across the world. However, hopefully you know that with every medication, there are risks, right? And so there are side effects of this medication. And some of those are metabolic, so increase in blood sugar. Maybe the insulin in your body that helps control blood sugar doesn't work quite as well. There's some changes or risks to potential increases in lipid values, as well as fatigue and even perhaps increased risk of infection. And so all these side effects come from data that are in patient populations that are using very high doses of the medication to treat their indication. And so we think, and there's evidence to suggest, that these higher doses are what is leading to some of these adverse side effects. So for the context of aging, we are significantly lowering our dosing protocols to try to minimize these risks for adverse events. And I'll try to expand on that a little bit. There's been some data to show that rapamycin can differentially impact 2 different complexes within this mTOR signaling pathway. And I'm not going to try to get into the weeds of this. I'll try to keep it very high level. But mTOR belongs to 2 complexes, complex 1 on the left and complex 2 on the right. So mTOR complex 1 generally increases with age. It's been associated with shortening of lifespan and health span. Whereas mTOR complex 2 has been linked to, it's been suggested that it's needed. That signaling pathway is needed to maintain health and function. And so when there's been studies in mice where they kind of genetically and specifically disrupt the mTORC1 signaling pathway, we actually show lifespan extension. But if you do the same thing in the mTORC2 pathway, there's increased metabolic dysfunction, frailty, and general poor quality of life. So this suggests to us that when we give rapamycin, we know that it can acutely and potently attenuate the mTORC1 pathway, but we know when it's given for prolonged periods of time at relatively high levels, it can also inhibit mTORC2. So we really think, based off of the data, that the function of rapamycin on mTOR complex 1 and attenuating that pathway is generally the positive aspect, so what we would call perhaps geroprotective. Whereas when we inhibit mTOR2 when giving these high doses for prolonged periods of time, that's one of the reasons why we may see these adverse side effects. So with that body of knowledge over the last decade, when designing trials to try to test this medication, we really are interested in identifying dosing strategies that will specifically inhibit the mTOR2 pathway without affecting the mTOR complex 2 pathway. So that way we can kind of capitalize on the benefits while minimizing the risks. And there's some data to suggest that we can actually do this. And so this is a study from almost a decade ago, and this is the study that showed improvements in influenza vaccine efficacy. And so what they did is they did different dosing regimens. They used a very low 0.5 milligram per day, 5 milligrams once per week, and 20 milligrams once per week. And what they showed is that the 5 milligram daily and the 5 milligram once weekly, so relatively low doses, they were actually effective at improving the titer response to the influenza vaccine in 2 of the 3 strains tested. That was their primary endpoint. And so basically, anything above this dark bar would indicate that it met its primary endpoint in 2 of the 3 vaccines. And the higher dose did not. So there's always this idea that maybe more is better, and that's not always obviously clearly the case here. In addition to showing that the improvements in vaccine efficacy, they did show that these lower daily doses that were effective at improving the immune response also targeted the mTOR complex 1 pathway, and there were very few adverse events. So no difference in serious adverse events versus the placebo, and the most common side effects that they observed were mouth ulcers, so like a canker sore, as well as increased triglycerides or LDL. And again, that was in very low percentage of the population, so 11% and 7% respectively. So overall, these initial data suggest that these low daily and weekly intermittent doses, they appear to be safe, fairly well tolerable, they attenuate the intended target, and they improve vaccine efficacy. So these are all really, really good things. And so one of the areas that we're interested is to expand this. What else can rapamycin potentially improve besides just the immune function? There have been a number of individuals across the country that are starting to take this off-label. Remember, this is not FDA approved, but there are people that have access to this. And so some researchers did a survey on some of these individuals who were taking rapamycin. So these are the rapamycin users on the left, and then non-users, basically meaning people who are interested in healthy longevity but aren't actively taking the drug. And overall, these are all self-reported documentation, but the rapamycin users suggested that they had decreased abdominal cramps, depression, muscle pain and tightness, anxiety. So a lot of, again, perceived benefits from people who are taking this off-label. Again, I don't recommend that anyone here start doing that, but this is the research to date. But what remains unknown? We know that greater doses have greater lifespan extending effects in animals, but we also know that higher doses also have greater risk profile in humans. And so when this drug came to market, they did a lot of testing in young men, and then they did a lot of testing in the patient populations that they were trying to bring this to market for. What they didn't do is really explore other populations. And so we really don't understand how the drug is moving through the body and impacting the tissues and the targets in older adults. So the pharmacokinetics is how the drug moves through the body. Pharmacodynamics is how it actually works in your tissues and cells. We also really have no real clear evidence yet if rapamycin can be effective to improve functional outcomes and perhaps extend healthy longevity in humans. And so much more studies and longer studies are going to be needed before we can actually make that determination. And so to help get us started on understanding whether there can be some benefits of rapamycin, we have a phase 1 trial ongoing right now. This is funded by the National Institutes on Aging, which means it's partially funded by your tax dollars. And so the objective here is really to identify whether, what is the safe and effective dose? That's really the take-home message. And so we want to identify that, and we do that by identifying whether people experience what are called dose-limited toxicities, and this seems scary, but we've picked a dose-limited toxicity defined as a grade 2 adverse event. And basically what that means is that this is something that you notice, but it doesn't necessarily impact your daily living. So it might be an annoyance, but not necessarily something that would prevent you from taking on daily life, right? And so that is important because these are healthy individuals, and we want to be conservative when we're talking about any potential risks. We're also going to be evaluating the pharmacokinetics and pharmacodynamics, and then for safety, understanding whether this drug can actually intervene on different biomarkers and biology of aging. So we have a suite of markers that we're going to look at that generally changes with age, and we want to see if the drug can help improve some of those biomarkers. And so we go through a very thorough screening with our medical monitoring team to ensure that everyone is safe to participate. So we screen out any sort of contraindicated medications as well as different conditions that would put people at greater risk. After that, there's a series of visits that occur, and we would be doing body composition. We'll be collecting different biospecimens such as blood, urine, and muscle, and also evaluating metabolic functions such as oral glucose tolerance test. We'll be doing that before and after a 6-week intervention, where people will be assigned to take either 5, 10, or even perhaps 15 milligrams once per week. And so I want to just share briefly of the study design of how this works. And so how we move through this study is informed on what we learn from the individuals that are actually going through it. And so that's the fancy way of describing this Boyne study design. And so this is hypothetical data. And so each box here represents a cohort of 3 individuals. So each dot is an individual. And the dose level that we propose is 5, 10, and 15 milligrams on the y-axis. And so in this scenario, we might have a cohort of 3 individuals where one person reports having a mild adverse event. And so based off of the guidelines that we have, that would suggest that we maintain the same dosing schedule for the next cohort. And since in this cohort there's also have one adverse event, then we would maintain it for the 3rd cohort. And as you notice, there's no dose-limited toxicity in the 3rd cohort, which is that dark circle. So now they're all open and clear. This meets our threshold of actually being able to safely escalate the dosing protocol for the next cohort. However, you can see that in this scenario, 2 people had mild adverse events. So we know that maybe we need to come back down. And so our protocol defines a dose de-escalation, so we come back down. And so this allows us, this is a newer study design that has been tested in the cancer field, so that way we can minimize the amount of risk that people are associated with taking too high of a drug dose, but we can also minimize the risk of people having too low of a dose where we won't see anything that's effective. So this is our way of hopefully maximizing the benefits that can come out of this study while minimizing risk. We've also done some pharmacokinetic analysis so far, and this isn't super exciting, but we see the expected curves. And the important thing is that we give this on a weekly basis, and the drug comes back down within 2 days, nearly identical to their resting value. And that is important because these resting kind of what we call trough values are related to the risk of adverse events. And so we are showing that we are having very low trough values of this drug 2 days after they take the drug and up to seven days after. So again, we seem to be doing everything in our power to make sure that we have the appropriate dosing protocols to minimize risk. During this study, we also use continuous glucose monitors, and this is a really fascinating kind of concept. So continuous glucose monitors are actually placed on the abdomen, and what they do is they can actually measure blood or glucose levels every 5 minutes throughout the day. You can wear them for 7 to 10 days. And what this is, why we like this so much is that when you take a fasting blood glucose measurement, you don't really get a picture of what's going on throughout the day. It's just a static snapshot at that particular time. And in our experience, when we take fasting blood glucose, a lot of times the people look identical. So they have very similar fasting blood glucose values. But what you can see here are 3 different participants that had the same fasting glucose but had very different glucose levels throughout the week, or throughout the day, excuse me. And so we think there's a lot of subtlety in here that will be informative on whether this medication can either improve or hopefully not worsen any sort of glucose changes throughout the study. And these are some of the preliminary data so far in a very small cohort of individuals. And it didn't reach statistical significance because the study is still ongoing, but we do see a general decline in some of the one indices of glucose variability, which is here. So this is a positive result. And then we don't see any sort of dysregulation in response to an oral glucose tolerance test, which is shown here. Seems to be no glucose intolerance, so no glucose risk factors there. We also have just been kind of questioning, we have a questionnaire to try to identify whether people are actually having positive kind of responses, their perception of their response. And so this is their report on emotional well-being. So this is improving. This is a good thing. So we see that the drug seems to be improving people's emotional well-being, or at least their perception of that. I will say that this should be taken with a grain of salt. There is no placebo, so there is a large placebo effect that can occur. So people believe that it is improving, and this has been shown in another study where on the right, it's a 5 milligram dose of rapamycin. There was an improvement in their emotional score, but right next to it, you can see that the placebo also resulted in an improvement in emotional score as well. So we are definitely taking this with a grain of salt. I think the positive here is that we didn't see it go in the opposite direction. So this is an ongoing trial, and we are actively recruiting and trying to engage people. And by the end of it, we think that through this collaboration with our medical monitoring team, who's a clinical nephrologist, we can identify a safe dose. With our bioinformaticists, we can identify what is effective at manipulating the biology of aging, and then we can use our pharmacologist to kind of integrate these data to identify the most safe and effective dosing scheme for older adults. And then last, we have an ongoing phase 2 trial. So it's a very similar scope, but it's a much larger study and longer study. So this is 24 weeks where people would receive either 0.5 milligram per day or 5 milligrams once per week, because this is what has been shown to improve vaccine efficacy and had very tolerable effects in a previous study that we just discussed. And so again, when they go through people will go through a very thorough medical examination to make sure they are safe and appropriate to participate. And then there are several different studies so that we can evaluate body composition, physical function and muscle strength, heart function and health, as well as brain function and brain health. And so we will do those before and after to see how this drug might improve or maintain those functional outcomes. And so here, this is kind of a different way of presenting that. So we are interested in 4 different really primary domains, or I guess 5. Our metabolic health, cardiac health and function, our physical health and ability, as well as our cognitive health and function, and then our safety metrics. So throughout this 24-week period, we routinely evaluate participants, identify any sort of adverse events, review their blood work on a monthly basis to make sure that people are safe throughout this study. For this, and we are going to do a whole suite of investigation in the biological aspect to make sure that we understand how the drug affects metabolites, proteins, genes, et cetera, all these things that become perhaps dysfunctional with aging, and see whether or not everolimus can help improve these outcomes. In this particular study, we are interested in older adults that might have slightly increased blood sugar or A1C. So these are insulin resistant to pre-diabetic individuals. And so the reason we are interested in this is because with these potential risk factors associated with impaired glucose and A1C, that actually increases the age-related conditions of type 2 diabetes, cardiovascular disease, and dementia. And so we think by targeting this population, we will be able to definitely, hopefully see improvements. And importantly, all these conditions have been delayed in animal models. So we have a data set to suggest that these can be improved in animal models, and now we're going to try to test this in humans. We also have a young control group. They don't receive the drug, but they do all the baseline testing, because the idea is if we think this is going to be an effective drug to improve the biology and the metabolism that we're measuring, we want to make sure that it's kind of reverting back towards a healthier person versus going the other direction. And sure enough, we have some preliminary data at baseline. I don't think these are necessarily surprising data, but we show that the older adults in our study have glucose intolerance. They have differences in heart function, suggesting that the older adults have lower heart function compared to the young, as well as skeletal muscle deficits in terms of power production. And then on the right is the MRI image, and I don't know if it shows up very clearly, but this is a representative image of a 28-year-old versus a 74-year-old. In the 28-year-old, you can see brighter colors. This is indicative of greater blood flow versus the 74-year-old otherwise healthy individual where there's more blue or lower blood flow. And so the idea is whether or not everolimus can kind of help improve these functional parameters to look more like the younger adults. Time will tell. And so in conclusion, one of the ideas is trying to identify different strategies to improve physiological function throughout the whole organism. And so that's one of the beauties of rapamycin, as well as exercise. It can affect many different cells and tissues in our body. And our working hypothesis is that using this drug to attenuate or inhibit the mTOR complex 1 pathway that we discussed is going to be beneficial while avoiding the risks associated with the mTOR2 inhibition. And so these are just our initial studies. And so I think from the information that we generate from the data we generate from these studies, it will really inform us on how to move forward with potential phase 3 trials. So really help us refine, improve, and determine whether or not there's data to support the idea that rapamycin could potentially extend healthy longevity in humans. So that is what I have. Our contact information, if you're interested in learning anything more about our studies, regardless if you want to participate, that's fine. Just if you have any questions, we'd be happy to address them. You can contact us here. Our study team is also here, and they have a booth in the back. So we have 3 coordinators from our team. They're waving their hands. So if you want to speak to them about the study at all, feel free to do that. They can address any questions that you might have. I want to thank the team. They do an outstanding job. They are extremely busy. They are wonderful at their jobs, and there's no way we could do it without them. I also want to thank the folks and collaborators on campus, because we have outstanding support to make sure that we maintain the safety and health of all our individuals that are participating in our study. And of course, you, for being here today to listen, to learn, and also contributing the tax dollars from the NIH to support these types of studies.